everyone, this is Adil. Today I am going to talk about basement membrane zone, which is also known as dermoepidermal junction. This junction is found between epidermis and dermis. As we know, stratum basale is the deepest layer of an epidermis, and in stratum basale there is the presence of keratin filament 5 and keratin filament 14, that is K5 by 14. These are keratin filaments K5 by 14, and these keratin filaments are attached to hemidesmosomes. These hemidesmosomes include prolactin, integrin, BPAG1 and BPAG2. These all are hemidesmosomal proteins. There is a layer which is found in the dermoepidermal junction which is known as lamina densa. This lamina densa is a matrix of collagen 4 and nidogen protein. Lamina densa and hemidesmosomes are connected each other by anchoring filaments. These green color filaments are anchoring filaments which anchor hemidesmosomes with lamina densa, and these anchoring filaments are made up of laminin protein, and this layer is known as lamina lucida or salt split layer. Why it is also known as salt split layer? Because it is the weakest layer in the dermoepidermal junction. Why it is weakest? Because if you take a section of a skin and keep this skin into the normal saline, then this skin will break from this lamina lucida. That's why this is the weakest layer in the dermoepidermal junction. There is another layer which is known as sublamina densa or lamina fibroreticularis, and this layer connects lamina densa with dermis, and in this layer anchoring fibrils are present which are made up of collagen 7. Defect in any of these proteins lead to subepidermal disorders. These are those disorders which are found below the epidermis. Now let's talk about subepidermal disorders. The examples of subepidermal disorders are bullous pemphigoid, dermatitis herpetiformis. Bullous pemphigoid and dermatitis herpetiformis are due to defect in BPAG1 protein, which is also known as BP230, because the molecular weight of the BPAG1 is 230 kilodalton. BP stands for bullous pemphigoid and AG stands for antigen. Bullous pemphigoid, linear IgA disease, lichen planus pemphigoidis, and herpes gestationalis are due to defect in BPAG2 protein, which is also known as collagen 17 or BPAG180, because the molecular weight of BPAG2 is 180 kilodalton. That's why it is also known as BPAG180. Epidermolysis bullosa dystrophians is due to defect in collagen 7. Here you need to remember a very important fact that bullous pemphigoid can be due to defect in BPAG1 or due to defect in BPAG2. A question has been repeatedly asked in various examinations that bullous pemphigoid is due to defect of BPAG1 or BPAG2. As we have earlier discussed that bullous pemphigoid is due to defect of both BPAG1 and BPAG2. If BPAG1 and BPAG2 are both given in the options, then mark BPAG2, because BPAG2 is more involved to cause bullous pemphigoid than BPAG1. Here is a question. This question has been asked in AIMS 2019, that arrange these disorders depending on their defects from superficial to deep. We need to arrange these disorders from superficial to deep, and the superficial disorder is pemphigus. Which is due to defect in desmoglin, and the desmoglin is present in epidermis. That's why the pemphigus is superficial. Epidermolysis bullosa dystrophians is due to defect in collagen 7, and collagen 7 is present just above the dermis. That's why epidermolysis bullosa dystrophians should be arranged as deep disorder. Then bullous pemphigoid, which is due to defect of both BPAG1 and BPAG2, but linear IgA disease is only due to defect of BPAG2. Therefore, bullous pemphigoid should be placed before the linear IgA disease. This is the arrangement of disorders from superficial to deep. The superficial disorder is pemphigus, then bullous pemphigoid, then linear IgA disease, and the deepest one is epidermolysis bullosa dystrophians. Here, point to be noted that bullous pemphigoid is the most common type of subepidermal disorder. Hope you have enjoyed this video. Thank you.